Yes. So in terms of how in vitro models will be potentially complementing or replacing in vivo models over time, I don't think we're at the stage of replacing at the moment, certainly, but I do think there's sort of two components, two considerations that we need to make at this stage. So one is the technology readiness of in vitro models. So how are they at the stage of providing the same amount of information as in vivo models currently do to help therapy developers make the decision about their compounds. And the second one is readiness of the regulators. So one is tech readiness and one is where the regulators stand. I think from the regulators' point of view, I'm sure everybody working in the field is aware that FDA is sort of continuously reviewing the approach to in vitro models, and there have been some modernization acts that passed through the Senate over the last couple of years. So clearly, regulators are on the side of that we need to be exploring in vitro models further. But on the other hand, it's about building enough data in collaboration between industry, academics, therapy developers, to really build a strong case that we are ready to make the jump, or at least to really substantially change how things are done currently. And I think we're somewhere at this stage now. So there's clearly a very strong case for in vitro models. There's clearly some real cases of companies using in vitro model data for IND submissions. But I think for us to really kind of push with the envelope, so to speak, we need to really focus on developing these data sets to sort of bulk up the validation data that we currently have on in vitro models. And that can't be done in isolation by one group or one company. I think it has to be done as a big collaborative effort. So the more of these collaborative projects we do, and the more we highlight the results to the regulators, the quicker we'll be kind of getting through to the stage where this landscape of preclinical testing changes. And just to add to that, the discussion we had earlier about AI and in silico modeling, I think it's also about integrating other technologies into the mix. So it's not just in vivo, in vitro, there's also in silico part. So it could be that by combining in silico with in vitro models, that adds additional sort of, it adds sort of a more strength to in vitro models, if you wish, to then also put them sort of higher up in terms of which models to choose in preclinical setting. I mean, we've been talking about sort of very complex models in the organ and a chip for a while. I think this sort of ability to mimic with high degree of similarity what's going on in vitro, sort of in vivo to in vitro, that trend sort of continues, and you can see a lot more complex functional validation of models in vitro that we probably didn't see even 3 or 4 years ago. But I don't think it's all about the complexity of the models, because at the end of the day, you just need to have the right model to answer your question. So although we are kind of moving towards more complexity, but I wouldn't say this is an absolute prerequisite. I think we're currently just looking at assessing complexity versus ease of data interpretation versus ease of use. And so I think we're kind of somewhere, we're still pushing the boundaries. There's some trends towards combining maybe different microtissue types into one, whether it's through microfluidics or other similar types of approaches to see the interaction of various mini organs with each other. So there's a lot more work going on in that area, but I suppose we'll wait and see where it takes us. I mean, for us, in terms of our approach to innovation, for us the key is constantly revisiting the role that we see for ourselves in the drug discovery process. What we aim to do is to give confidence to customers when making decisions about their drugs. So it is finding the right model for the right application. So working closely with our customers, working closely with the regulators, working closely with key opinion leaders. So really trying to make sure that whatever the decision we make, whether it's to do with innovation in our processes or our models, that it's going to be right for the applications that the models are being used for. So we innovate both on the side of processes and on the side of models. So it's not just, having the best in vitro model, but how do we actually approach, whether it's to do with how we run our operations, how we run our customer projects. Yeah, we innovate across all areas, and it's based on the feedback that we get externally. I think the point I'm trying to make, I think now a lot of it is around data. You can utilize data a lot more than you could maybe even 5 or 10 years ago, even 5 years ago. The amount of data we can get is huge. I think one of the greatest impact is that it's an additional sort of tool in their toolbox, which potentially might replace what they're currently using. So it's about its predictive value. So in vitro models, in many cases, are already having a higher predictive value than a lot of the animal models, especially when it comes to gene therapy. Some animal models just simply don't exist, so you won't be able to test the exact therapeutic vectors that have been tested in human in vitro model in an animal model. So that's a pretty clear case for direct impact on how customers approach their preclinical testing. And obviously, with the models having high predictive value, it sort of facilitates the decision making about which lead candidates to take through to the next step. I think in terms of us keeping innovating in the field, we need to bring it back to the customer, but also to the patient. So it's our interest, it's in our interest, and in all of our interest is to get the drugs out to patients as quickly as possible. And some of these patients don't have the time to wait, because a lot of the diseases that our customers are working on and targeting are fast degenerating. So if we can provide them a model which has higher predictive value and gives out robust data, which then de-risks the next steps in the drug discovery process, meaning, you know, if this data set allows somebody to make a decision that yes, it's ready, you know, it's going to really strengthen the case for taking the drug to the clinic and speed up the process. I think that's the key value. How quickly can the drugs get to the patient? And I think here the stakeholders are both customers and patients, and we shouldn't forget about that. So I don't think in vitro models will completely replace in vivo models in the next 5 to 10 years. I think it's important to keep this at pace, this change. Because, you know, the risk of not using the right model is quite, I'm trying to say, you know, we don't want to say, you know, this is the goal to completely replace animals in 5 years time, because we need to go with the pace of the technology. So I don't think the in vitro models will replace animal models. I think they might replace them in certain scenarios, let's put it that way. There might be cases where animal models are just not available, in which case, yes, there will be an in vitro model replacing the animal model in that particular scenario for that particular question. And as I've said earlier, I think the next 5 to 10 years are going to be really important to build up enough data to give us confidence that maybe further down the line in the future, we may see a case where animal models are reduced even at a greater pace. So it's about generating validation data by forming sort of strong collaborative networks. So I think that's where we're at right now.